In this last video for Chapter 9, we're going to discuss one last thing, osmotic pressure. So osmosis is the process by which solvent molecules can pass through certain membranes, but solute particles cannot. And there is a tendency for solvent molecules to move from a less concentrated solution to a more concentrated solution until the concentrations are equal. And this tendency is called osmotic pressure. So this does have to do with concentration again. So down below I have a figure showing the process of osmosis. So let's start on the left side. We have a beaker, and this beaker has a sugar solution in it. And down the middle of the beaker is something called a semi-permeable membrane. So we've got the semi-permeable membrane. Now, what is that? So a semi-permeable membrane just means that certain molecules can pass through that divider, but other molecules cannot. And in this case, water can pass through that membrane, but sugar molecules cannot pass through it. All right, so on the left side of this beaker, we have a bunch of water molecules which are represented by blue dots, and we have one sugar molecule. On the right side of the beaker, we have water molecules as well, and we have 4 sugar molecules. So the right-hand side looks like it's more concentrated. It has more solute per volume of solution. And the left side is less concentrated. Now, with osmosis, remember, solvent molecules are going to move to the side that's more concentrated until both sides have equal concentration. All right, so the way it is now, they're not equally concentrated, right? The left side is less concentrated, the right side is more concentrated. So let's just write that. Let's see, I'll write up here, this is more concentrated. So through the process of osmosis, water is going to actually move towards the right-hand side until we get the figure on the right here. So the volume of water decreased on the left and increased on the right. So we actually diluted the right-hand side of the beaker until the concentration of the left-hand side equals the concentration of the right-hand side. So even though we still have 4 sugar molecules on the right and only 1 on the left, we did add more water to the right. So we basically found equilibrium between the two sides. So we created a situation where the concentrations are going to match. All right. Now, what is osmotic pressure? Well, we can consider that in terms of how much pressure we would need in order to go back to the left-hand side. So imagine we have a movable lid here, and the pressure we would need to push this water back through to the left is the osmotic pressure. So external pressure can be exerted on a solution to counter the flow of solvent, so to stop the flow or to even push it back the other way. So the pressure that's required to stop the osmosis is equal to the osmotic pressure of the solution. All right, so let's go to our actual term for this pressure, osmolarity. or we could abbreviate that as osmol. So this is a way of reporting the total number of particles in a solution so that we can determine the osmotic pressure of a solution. So osmol or osmolarity is just equal to the concentration of the solution, so moles per liter, times this expression I. Now I is the number of particles a formula unit of the solute makes when it dissolves. So, for example, previously we saw that salts can dissolve in water. And we saw that water is actually going to break apart the ionic compound, the salt, until it's surrounding each individual ion. So, if we dissolve sodium chloride in water, sodium chloride is going to separate into sodium ions and chloride ions. So we don't just have NaCl floating around in water, we have individual sodium ions floating around and individual chloride ions floating around. So we've actually doubled the amount of ions that are floating around in solution, right? So this osmolarity value I is equal to 2 for sodium chloride. Now, what if we had something like magnesium chloride, MgCl2? Well, if this is dissolved in water, we get a magnesium ion and 2 chloride ions. So the value of I for magnesium chloride would be 3. So it just depends on how many ions you have per formula unit. And remember, formula unit is how we refer to an ionic compound and its simplest ratio of ions. So let's do an example problem where we use this osmolarity. So let's say we have a 0.5 molar sodium chloride solution and a 0.3 molar calcium nitrate solution. And they're placed on opposite sides of a semi-permeable membrane. So let's draw a picture because I think that's always helpful for these types of problems. So we've got our beaker, we've got our semi-permeable membrane. And remember, the semi-permeable membrane only allows water to pass through. So the solute, the ionic compounds or the ions, they cannot pass through that membrane. All right, and then we've got our water. And on the left-hand side, let's put our sodium chloride in there. So we've got sodium ions and chloride ions floating around in solution because they've already dissolved. And then on the right-hand side, let's place our calcium nitrate. Now notice that we have 1 calcium and 2 nitrates. So when calcium nitrate dissolves, we have a calcium ion and then 2 nitrate ions. All right. So let's determine the osmolarity of each solution, and then we'll predict the direction of solvent flow. Okay, so remember that osmolarity is equal to the concentration times I. So for sodium chloride, we already said that I is equal to 2 because that sodium chloride is going to break apart into 2 ions. What about for calcium nitrate? What would I equal for calcium nitrate? 3. Since calcium nitrate broke apart into 3 ions. All right, so Let's start with sodium chloride. Let's calculate this osmolarity. So all we have to do is multiply 0.5 moles per liter times 2. So remember the concentration for sodium chloride was 0.5. All right, so that gives us, let's see, a value of 1. Super easy, right? Okay, now let's look at our other solution, calcium nitrate. So we're just going to multiply its concentration by 3, since I is equal to 3. So 0.3 times 3, that should give us 0.9. Okay. So it looks like the osmolarity of our sodium chloride solution is higher. So that is more concentrated, right? So that means that water is going to flow from right to left until we reach equal concentrations on both sides. All right, so the solvent will flow to the left. According to our picture, obviously. If we had swapped it, it would be opposite. All right, so there's one more problem that I want to go over. So let's say we have a 1.5 molar sugar solution. So C6H12O6 is sugar. And we have a 0.4 molar solution of aluminum nitrate. And again, they're placed on opposite sides of a semi-permeable membrane. So let's determine the osmolarity and predict the direction of solvent flow. So let's draw a beaker again. And we'll put our semi-permeable membrane down the middle. And then we'll draw our water. And let's put our sugar solution on the left. Now, sugar is not an ionic compound. So this, when it dissolves, it's not going to break apart like an ionic compound. It's just going to stay intact. So if it doesn't break apart, what would the value of I be? Just 1. So if something doesn't break apart in solution, if it just stays the same, its value of I is 1. Now on the right-hand side, let's put our solution of aluminum nitrate. So notice there's 1 aluminum and 3 nitrates. So when that breaks apart, because it is an ionic compound, we'll have 1 aluminum ion and then 3 nitrate ions. So what is the value of I for aluminum nitrate? 4. All right, so see if you can calculate the osmolarity for our sugar solution, and then see if you can calculate the osmolarity of the aluminum nitrate solution. And then try to predict which side the water will flow to. So pause the video, try it on your own, and then we'll go over it together. Okay, so for our sugar solution, this is going to be super easy. We just multiply the concentration we were given, 1.5 moles per liter, times 1. So that just stays the same, 1.5. All right, and then let's do the same for aluminum nitrate. So the concentration we were given is 0.4 moles per liter, and we're going to multiply that by 4, since that's our value of I. Let's see, 0.4 times 4. That's going to be 1.6. Yeah, 1.6 moles per liter. All right, so it looks like our aluminum nitrate solution is more concentrated in this case. So water is going to flow to the right, towards aluminum nitrate. And then water will keep flowing until we hit that point where the concentrations are the same on both sides. So in this problem, well, actually, in the last problem, right away it looked like sodium chloride was more concentrated, and that was still true when we calculated the osmolarity, right? But in this second problem, initially it looked like the sugar solution was more concentrated than the aluminum nitrate solution, so it had a higher concentration. But when we calculated the osmolarity, we found that the aluminum nitrate solution was more concentrated, especially once those ions broke apart in solution. We had more solute. All right. Now, how can we apply this to the real world? Well, one application of osmolarity and osmotic pressure is dialysis. So our kidneys actually filter our blood to remove any waste and extra water from our system, and then we expel all of that as urine. Now, there are some diseases that will cause the kidneys not to function properly, and so we end up with a buildup of waste in our bloodstream. Now, if one way to solve that is to perform a kidney transplant. However, if that's not available, or if there's reasons why that wouldn't work for the patient, then we can use dialysis. And dialysis will help remove waste and excess water from our blood. So you might have heard of hemodialysis. This is where a patient's blood is passed through a tube, and this will travel through a machine that acts as a kidney. So it's an artificial kidney. And this is also called a dialysis machine. And then there's a section of tubing that has a semi-permeable membrane, which is immersed in a solution of sterile water, glucose, amino acids, and electrolytes. And amino acids are what make up proteins in our body. So the osmotic pressure of the blood in the tubing will force waste molecules and any excess water through that semi-permeable membrane into the sterile solution. And then after that's cleaned, then the blood goes back to the body. So this is a really cool application, and it helps a lot of patients. Now, another application, if we think about our cells in our body, the solution around our cells can have a huge impact on whether the cells are normal, or if they might shrink, or if they might expand. So in our cells, which you can see down below, we've got our cell here, our red blood cell. The cell wall is made up of a semi-permeable membrane. So only certain things can pass through that semi-permeable membrane. And the osmotic pressure of our bodily fluids will have important consequences on that red blood cell. So for example, Let's start with this isotonic solution. So an isotonic solution just means that we have the same concentration outside the cell and inside the cell. So we've already reached osmosis. So water can pass in to the cell and out of the cell, and everything just stays normal. However, if you have a hypotonic solution surrounding the cell, that means that the surrounding solution is actually less concentrated. Less concentrated. And the inside of the red blood cell is more concentrated. So just like we saw before, if something is more concentrated, water is going to flow into that part of the system. So if water is flowing into the red blood cell, eventually that red blood cell is going to swell. And sometimes it can even burst if the pressure becomes too much. And then if you have a hypertonic solution, that means that the surrounding solution is more concentrated. And the red blood cell is less concentrated. So water is going to flow out of the red blood cell and into the surrounding solution, and that means the red blood cell is going to shrink. And we actually call that a crenated red blood cell. I think I pronounced that right. I'm not a biologist, so correct me if I'm wrong. All right, so this can have a huge impact on our cells, and that's why you always want to drink enough water to keep your fluids at a normal concentration. So let's say you're really dehydrated, that could cause a hypertonic solution. Or if you eat too much salt, that could cause a hypertonic solution. So you always want to try to keep your levels somewhat consistent. Now, I mentioned that those cell walls are made of semi-permeable membranes. So I have a diagram here kind of showing that cell wall, that semi-permeable membrane. And the semi-permeable membrane acts as a barrier that separates the inside of the cell from other cells and from the solutions around them. Now, sometimes this cell membrane is called a plasma membrane. And again, it's semi-permeable, which means that some things can enter the cell and exit the cell, and some things cannot. So water can pretty easily go in and out. But notice that within this cell wall, we have these things that are kind of within the cell wall. So those are typically proteins that can allow solute to pass through. So things like sodium ions, potassium ions, and other solute particles that are in our system. And then notice too, we have these kind of little fiber looking things. So these are what make up the cell wall. These are lipids, and you're actually going to learn more about this if you take Chem 131 next. You'll go into more detail on what those membranes are made up of. And if you take more biology classes as well, you'll probably hear more about this then. All right, so that's the end of chapter 9. I know it was a lot of information, so feel free to reach out to me if you have any questions, and I will see you in class.